Welcome. Today we are diving into meiosis, the remarkable process that turns ordinary diploid cells into specialized gametes. This process is essential for sexual reproduction, which means no meiosis means no genetic variation equals a very boring world. Meiosis creates diversity through crossing over and independent assortment. Two mechanisms that students often hear about but don't fully understand. By the end of this lesson, you'll not only understand them, you'll be able to explain with confidence on AP and IB exams. Think of this video as a guided tour through cellular matchmaking. Let's get started. Meiosis matters because it creates gametes, eggs and sperm, each containing half the normal number of chromosomes. This reduction is crucial because fertilization restores the diploid state, preventing chromosome overload. Without meiosis, organisms would double their chromosome count every generation, which would get ridiculous very fast. Meiosis also creates variation by shuffling genetic information. This variation is the fuel that evolution runs on. So, when your biology teacher says meiosis is important, they're not exaggerating. It's literally the reason species survive long term. Before we dive into meiosis, it's essential to learn the difference between diploid and haploid cells. Diploid cells contain two sets of chromosomes, one from each parent, while haploid cells contain only one set. Gametes are always haploid, so that fertilization restores the diploid state. Somatic cells, like skin or muscle cells, are diploid because they aren't involved in reproduction. Keeping this distinction clear will help you understand why meiosis needs two divisions. Think of it as splitting a complete set into two balanced halves. Meiosis consists of two divisions, Meiosis I and Meiosis II. During Meiosis I, homologous chromosomes, pairs you inherit from each parent, separate. This is the major chromosome reducing division that turns diploid cells into haploid cells. Meiosis II resembles mitosis, separating sister chromatids. Many students confuse these two phases, but remembering 1 equals homologs and 2 equals chromatids makes the difference clear. Let's break each stage down step by step. Prophase I is the most important is one of the most important steps because crossing over happens here. Chromosomes condense and homologous chromosomes pair tightly, forming tetrads. This pairing allows sections of chromatids to exchange genetic information. Crossing over generates brand new allele combinations not found in either parent. This is why siblings look similar but not identical, except for twins who beat the system. Understanding crossing over is essential for AP and IB genetics questions. During metaphase, homologous chromosomes line up along the equator of the cell. The key idea is that their orientation is random. This randomness is called independent assortment. It means the maternal chromosome might go to one side, or the paternal might. There is no pattern. With 23 pairs in humans, this creates over 8 million possible combinations before crossing over is even added. It's nature's version of shuffling a deck of cards, but with far more outcomes. In Anaphase I, homologous chromosomes are pulled to opposite sides of the cell. Unlike mitosis, the sister chromatids stay together at this stage. Telophase I then forms two haploid cells, meaning each now contains one chromosome from each pair. This is the division that officially reduces chromosome number. Many students mistakenly think the cells are diploid, but nope, this is the haploid checkpoint. Cytokinesis follows, creating two distinct cells ready for meiosis II. Meiosis II is structurally very similar to mitosis, but with haploid cells entering instead of diploid ones. The sister chromatids line up again and separate. This ensures each haploid gamete receives only one chromatid from each duplicated chromosome. The process is efficient and mirrors what you've already learned about mitosis, making this division easier for students to understand. When complete, the result is four genetically distinct gametes. Each one is unique thanks to crossing over and independent assortment. Meiosis and mitosis share similarities but serve entirely different purposes. Mitosis creates identical copies used for growth and repair. Meiosis, however, produces gametes for sexual reproduction. Mitosis results in two identical diploid cells, while meiosis produces four genetically varied haploid cells. On exams, this comparison shows up constantly, so it's worth memorizing. Whenever in doubt, think mitosis makes clones, meiosis makes diversity. Meiosis boosts genetic variation in three major ways. First is crossing over, which physically swaps DNA segments between homologous chromosomes. Second is independent assortment, which mixes maternal and paternal chromosomes in many combinations. Finally, random fertilization amplifies variation because any sperm can fertilize any egg. When combined, these mechanisms create enormous genetic diversity. This is why you and your sibling don't look like photocopies of each other, unless you feel emotionally like the forgotten middle page of the printer. AP and IB exams frequently test meiosis in both multiple choice and free response questions. You'll clearly need to distinguish meiosis I from meiosis II and explain how variation arises. Comparing mitosis to meiosis is also a major theme. Being able to draw simple diagrams is extremely helpful, especially on IB paper responses. Practicing, describing, crossing over, and independent assortment in your own words will be very helpful. If you can teach meiosis out loud without panicking, you're ready for test day. That's it for now. Don't forget to subscribe, like, and check out Dr. B's tutorials on Amazon for full AP and IB study guides. See the first link in the comment below. For more tutorials and test prep videos, subscribe to our YouTube channel, Dr. B's Tutorials.